So hello everyone. I'll try to also be on time, but I don't promise it because it's the first time I present mostly the techno that we develop, and I usually focus on the biology. So in that talk, I'll discuss the broad topics that we study in the lab, the kind of tools that we build to tackle these questions, and I'll give you some hints of the biology that we study. So the big question that we are interested in is confined growth. So cells, when they proliferate, they grow, they expand in volume, and then they divide, and that leads to the multiplication of cells. But the thing is that in most of the cases, and that's actually more the rule than the exception, cells proliferate in a confined environment. So they are surrounded by something, by the environment, by other cells or whatever. And for cells to keep on growing, they need to push against their surroundings in order to increase their volume, such that at the population level, there is the emergence of a type of mechanical stress that we call growth-induced pressure. It's compressive for the cells, it's compressive for the surroundings, and that is a necessary condition for cells to keep on growing when they are spatially limited. So you can also find this sort of growth-induced pressure if you just think of a cohesive assembly of cells. If you look at the cell at the center, for instance, surrounded by other cells, it's bound to the other cells. So in order to grow, it will need to push against these adhesive properties such that growth-induced pressure will also emerge. So this type of mechanical stress, you find it everywhere in the living, and that ranges from plants to mammalian cells to bacteria to fungi. And you have it both in physiological conditions, that's the blue, and or in pathological conditions. So I recently wrote a review on that. If you want to have more examples, I will not go through all the examples, but basically for plants, you can just think of the roots of trees sprouting into the ground. And when they sprout into the ground, they don't have the space to grow there, so they will build up an enormous amount of force. That is growth-induced, actually, and that's enough to break concrete, for instance. A more famous case, maybe, that we study a lot in the lab also is the case of tumors. So solid tumors, they grow in the body of an organ, and they grow where there's no space initially for them to grow. So when they grow in an organ, they will build up growth-induced pressure that is in the kilopascal range for animal cells, and that has been experienced in vivo, ex vivo, sorry, measured ex vivo. If you take out the tumor from the body and you put just what you call a biopsy punch, basically a big cylinder, you drill a hole, and then what you can observe is that the tumor relaxes, and that's basically a sign that there was stored stress inside the tumor. And from the reconstruction and reverse engineering, knowing the mechanical properties of the tumor, then you can come back to the pressure that was stored inside the tumor, and that's in the kilopascal range. So the big question is, how do we study growth-induced pressure? So that's where microfluidics enters. The first type of microfluidic device that I developed was for a fungus that's called Saccharomyces cerevisiae, also the baker's yeast. So that yeast, you can find it anywhere in the living. You find it in beers, in wine, in bread, in life, basically. And it's a perfect size because it's 5 micrometer, which means that it's good enough for classical photolithography. And also the type of mechanical stress that it builds up is in the hundreds of kilopascal range, meaning that it can deform classical polydimethyl siloxane, which is the material that we use. So the devices that we build are always the same ones. So here you have a schematic. So what we have is a cage where we put cells in through an inlet. That's also an outlet. It's kind of a valve that we can close. And this chamber is connected on the side to a set of narrow channels that are too small for the cells to go through, but big enough so that we can set the chemical environment. So basically, we can work at a roughly constant chemical environment, and I'm happy to discuss that at the end of the talk. And basically, we just let the cells grow in this device. They build up mechanical stress, and in that case, we can just see and measure the mechanical stress by the deformation of the PDMS. So how do we trap the cells practically, and how do these valves work? So that was the work of my postdoc. What we see is that these microorganisms, fungi, they're not super deformable. So because they're not very deformable, what we can use is actually the fact that they will form force chains inside the chambers that span through the whole chamber, and they will jam. So if you play with the geometry of the inlet, if you basically change the angle, then you will basically enhance the propensity of the cells to build force chains, such that at a critical angle there that you can see, there's a massive buildup of force chains and the cells are fully blocked. Here you have a simulation of the force chains, and here you have experiments where pressure builds up, and then you have poof, an avalanche, basically because the pressure is higher than the force chain, and then the cells escape the chamber. So we can play with that and basically also play with the amount of mechanical stress that this fungus can build. Okay, so then if we want to study that in different organisms, how do we do? So over the years, we built different devices to work on smaller organisms, bacteria, and on bigger organisms, which are mammalian cells. So for smaller organisms, for bacteria in particular, so bacteria are submicrometric in their smaller size. They're basically 0.5 micrometer. So the main challenge was that we needed to downscale a lot the macro channels to get to nano channels, basically. And here we used, in combination or depending on the type of devices, we used one or the other one. We used either the stepper technology, which is basically a way to zoom and to project a shadow of what you want to design onto a mask, or e-beam lithography, where you can go very, very small in terms of resolution. And we built very small, 400 nanometer, we even went down to 300 nanometer channels that are macrometric in length, and that we were able to mold in PDMS. So with these devices, we were able to trap E. coli, Escherichia coli, which is a small bacterium. And these bacteria, they also build up large enough mechanical stress so that we can still use PDMS. They're also rigid, so just like yeast, we can also use the same type of valves that we have designed to jam the cells and to let them build up mechanical stress. The problem was actually when we wanted to study and adapt that to animal cells. So mammalian cells, in contrast to microbes, they don't have a cell wall, so they're not very rigid. They're actually highly deformable. And also the amount of pressure that they build up is much, much smaller, such that just relying on the raw deformation of PDMS wouldn't work to measure mechanical stress. So we had to basically get around these two problems. So first of all, we designed a pressure sensor in order to measure the force that these cells could develop. And we got inspired by macropillars technology, which are high aspect ratio pillars that are highly deformable, even if the material is super rigid. So basically what we built is a suspended membrane that is then highly deformable. Here you have a COMSOL simulation to calibrate the system. And then when the cells build up pressure, they can now deform highly. You see here it's macrometers, so 20 to 40 micrometer deformation for small pressure. And then we are able to measure the pressure. So that was not very complicated. The complicated part was actually to trap the cells. So to trap the cells, we developed what we call sliding elements. And for that, we got inspired by the work of Bastien, who's your host today. And the idea was to build macrofabricated structures that we can insert inside a PDMS chip to bring novel functions, or to also trap devices, to trap biological samples. So here, that's one example of how we do them. We can basically use dry film lamination, then do classical photolithography, and then detach the sliding elements to create them by the hundreds, and then we can insert them inside microfluidic devices. And the type of microfluidic that I will present is basically one where we have the biological sample. Here, the sliding elements, they have micro slits to feed the cells, and we can load the sample by the top and then slide the sliding element to then trap it. So here you have examples of the type of organisms that we can trap inside it. We can trap, of course, mammalian cells, but we can also trap living organisms, such like a nematode, Cenorhabditis elegans, or even the leg of the fruit fly, the Drosophila melanogaster, which we use also to study its development. So combining these two technologies, we are able to trap mammalian cells, and then they are fully confined, and then they will start to build the pressure, and they will deform this deformable element there, and they will build the pressure in the kilopascal range. In the next two slides, I want to give you two examples of what we do with these sliding elements as well in the lab. So the first thing that we do with these sliding elements is to adapt them to do macropipette aspiration on chip. So for those who don't know macropipette, it's a technique that is widely used in biology to characterize the rheological properties of cells or assemblies of cells, just like that, what we call an aggregate of cells, or even biomimetic samples. So the idea is that you basically trap this object in front of a cylinder, then you create a difference in pressure, and that object is aspirated. And based on the dynamic of aspiration of what we call the tongue inside this macropipette, you can extract its elasticity, its viscosity, and all sorts of rheological parameters. So here we can trap them, we can parallelize and have up to 25 spheroids, in that case, in parallel. But because we build these devices from the top down, we can also tune the geometry of the macropipette to have it like conical, for instance, if you want to study other types of properties. Another type of application that is unpublished is also going towards implantable electrodes on chips. So most of the time when you want to do electrochemistry in a microfluidic device to, for instance, I don't know, measure pH or oxygen or these sort of things that are super important in biology to measure metabolic activity, what you do is that you pattern the cover slip onto which you bound the microfluidic device. What we wanted to do is, and then basically when you use a microfluidic device, then it's throwable and you throw it and then you poof, you're done, and that's the end of it. We wanted to use that technology, the sliding elements, to combine them with electrode deposition in order to create electrodes on these sliding elements that you can then insert inside a microfluidic device in order to measure different sort of things. So here I give you a proof of principle that we have recently, finished, and that we are writing the paper of on a macro sliding element with which we can measure pH. Something also that you can see is that now these sliding elements, we make them in glass. That's also a new amelioration that we have. And for that, we use selective laser etching. For those who don't know that technology, I highly recommend that you have a look at it. It's a very, very interesting tech with a high resolution to structure glass, which you can use then to mold PDMS onto it, or also to create these sliding elements in a very effective and also cool material that is glass that is very easy to use and to pattern afterwards. All right, so with that tech introduction, what do we do with these devices? And because I'm sure you're dying to know what happens when cells are confined in terms of their biology. So the first thing that I want to show you is the pressure that different organisms build as a function of time. And here I show you pressure curves for mammalian cells, for the fungus Saccharomyces cerevisiae, but also for the bacteria Escherichia coli. What you can appreciate from these different curves is, first of all, the difference in the orders of magnitude at the pressure that these organisms build up in terms of pressure. Here, cerevisiae can build up to 1 megapascal, which is gigantic. It's highly challenging, actually, in terms of the binding of PDMS devices. And it's in the hours kind of time scale. If you go down to E. coli, it builds up much less pressure in the hundreds of kilopascal, and that's much rapid, quicker rise of the pressure in the hours, compared to also animal cells, which build pressure in the kilopascal range in terms of days. Basically, the pressure buildup is related, because it's growth, the pressure buildup is related to the division time of the organism. E. coli divides super quickly, and mammalian cells divide super slowly. And the pressure that these organisms develop is also related to their own mechanical properties. So for instance, Saccharomyces cerevisiae and E. coli, as I said, they have a rigid cell wall, and they use it to also have a very high internal pressure that we call the turbo pressure, that is used also for their everyday life, compared to animal cells, which don't have a rigid cell wall, meaning that they can't also sustain such a high pressure. And the second thing that I want you to notice is that these pressure curves, they are all concave, if you look at that. And basically what that means is that there's a slowdown of the processes leading to the buildup of pressure. And because buildup of pressure is growth-induced, it means that there has to be a decrease in growth as pressure builds up. And that we observe in almost all organisms. So what we can measure is the rate of growth, so the rate of volumetric increase of the cells as a function of time. For these different organisms as a function of time or pressure, it's basically the same. And what you can appreciate is that in all the cases, growth rate goes down. So I'm saying all with an asterisk because it's biology, so there's always a lot of counterexamples. For instance, we found instances where rate can first increase and then decrease, but that's a kind of a biological feedback that we can discuss also at the end of the talk if you want. But basically in almost all studied organisms, when pressure rises, growth rate decreases. So we were interested in why growth could be decreasing. And for that, we started to get interested in the amount of proteins, the biomass that cells produce as a function of time and pressure. So you can use a lot of different reporters in cells. For instance, we use that one, which is inducible. So basically when we induce through, in that case, starvation, that promoter, then the cells, they will accumulate a fluorophore, meaning that they will become more and more fluorescent. And then we can record the fluorescence intensity as a function of time at the single cell level. And that's the kind of curve that we have. And you could appreciate from the movies that when the cells are under pressure, they accumulate the proteins, the biomass, much slower than when the cells don't have any pressure. And that's very important for us because for cells to grow, they need to accumulate biomass. So it means that the decrease in growth that we observe is basically related to a decrease in biosynthesis. So we can model this rise in fluorescence intensity. I won't bother you with the details unless you're interested. But basically what we can extract is an effective protein production rate as a function of pressure. And that we can do again for all the organisms. And what we have shown is that there's some sort of universal decrease in protein production as pressure builds up. And again, there's an asterisk because for the organisms where we see a feedback where growth first increases and then decreases, we see exactly the same feedback in protein production. Protein first increases, then decreases. So we are now actively working to understand the origin for the decrease in protein biosynthesis. And we have a lot of ideas of what's happening. But one thing that we had in mind is that if this process is conserved for these very different organisms, from bacteria to fungi to mammalian cells, which have diverged in terms of evolution for hundreds of millions of years, there could have been something that is physical encoded in them that could actually explain that. And that's what we wanted to study. So in the last 2 slides, I think, of the talk, what I want to discuss is a physical property of the cell. And I'm sorry, it's not microfabrication, or it's microfabrication at the level of the cell. That is called macromolecular crowding. So that the idea of that property of the cell is that the cell is highly packed with macromolecules. The cell, inside the cell, is not empty. You have hundreds of grams per liter of macromolecules. And try to put, for instance, 300 grams of sugar inside 1 liter of water, and you will see that it's highly viscous. So that's about how viscous the cytoplasm, the inside of the cell, is. And these macromolecules are very important because they modulate the biochemical reaction rate inside the cell. If you increase a bit the crowding, the macromolecules around 2 substrates that will react, then you will first favor, for entropic reasons, the biochemical reaction. But then if crowding starts to be too high, then the diffusivity of these 2 substrates will start to be too low, and the reactions will then decrease very strongly, such that for any biochemical reactions inside the cell is an optimum. So what we hoped to find is that when the cells are confined, maybe everything slows down because the cells become more crowded. But how to study crowding? What we have built recently is nanoparticles that are genetically encoded inside the cells. What we mean by genetically encoded is that you don't need to inject them inside the cells. You basically, what we do is that we encode inside the cell a protein that can multimerize to form a multimer, and we fuse that multimer to a fluorescent protein, such that when we put that inside the cell, this genetic sequence, this genetic sequence is expressed, we create that monomer inside the cell, and that monomer then multimerizes to form this type of nanoparticles that we can reconstruct through electron tomography that are 40 nanometer for that particular particle, and that are super, super, super bright because they have, for that 120 mer, so they have 120 fluorescent proteins. We can target them in different subcellular compartments, and they are the size of a lot of relevant physical, biological complexes inside the cell. So what does it look like? It looks like this. Here you have these nanoparticles, they move. It's real-time movie, it's in an animal cell. So that's basically how fast things move inside you, inside the cells. And then you can track them, and through the tracking, you can extract a wealth of information, such as the diffusion coefficient. And what we see for Saccharomyces cerevisiae, when you look at the movies, I don't know how laggy it is for you, but what you could see, if it's not laggy, is that the nanoparticles, they diffuse much, much slower when the cells are under pressure. We can measure the diffusion coefficient, again, for all of the organisms. And what we see is that as pressure rises, there's a massive decrease in the diffusion, meaning that the cytoplasm, the inside of the cell, is becoming more and more viscous. And this time, there's no asterisk. That we find for any organism that we study. So as the cells are under pressure, they are more viscous. And because they are more viscous, biochemical reactions go down. So, and that will be my last slide. That's the message I want to convey of how cells grow when they are confined. But to understand how they grow when they are confined, we need to understand how they grow when they are not confined. So when cells grow, they accumulate biomass, that would be the big blobs, and osmolytes, that would be the small red dots. Osmolytes are essential for cells to swell through osmotic differences, such that water can go in. And osmolyte and biomass production have to be proportional somehow in order to keep the intracellular concentration, intracellular density constant. So when you increase biomass and osmolyte, then the cell will dilute through water increase, and then the concentration will remain constant. But when the cells are confined, what happens is that the cells accumulate still proportionally biomass and osmolyte, but the cell volume cannot increase because the cells are confined. So it means that the osmolyte concentration will increase, and that leads to pressure buildup, osmotic pressure buildup inside the cell, and that's the pressure that we measure. And at the same time, they accumulate biomass, and that leads to the increase in crowding in the viscosity that we have measured. And both these things are proportional, and that's why what we found, and that's why we see that as pressure builds up, biomass builds up, reaction rates slow down, and then biomass production slow down. So that's the main take-home. I won't give you a take-home message in terms of microfabrication, because I mean, I guess that you know super well, and you have already a lot of things. But the main take-home message of what we find is that when there's biomass production, that leads to crowding, and that feedbacks onto biomass production, slowing processes. So with that, I'd like to thank the members of the team and the past members that have significantly contributed in terms of the macrofabrication. And I also want to give a special thanks to the team. So that's the team is the team is the team of the clean room at LAAS-CNRS, and in particular David Bourrier for the SLE, Laurent Mazing and Fabien Minigante for the photo and nanolithography. And I thank you for your attention.